Okay, guys, we will make a start now. So welcome everyone to our A Level AQA Biology session. In today's lesson, we will be covering biochemical tests, which is the last part of the spec for carbohydrates. We looked at carbohydrates in our last lesson. Okay, so to start off with, what we are going for through in today's lesson, we're going to look at the test for starch, protein, and lipids, and for reducing sugars. That should be a nice recap from GCSE. And then there's one new part of the spec, which is testing for non-reducing sugars. Okay, if you haven't already, please confirm your attendance to the session by scanning the QR code which is on the screen, or you can click on the link which is in the Q&A. Okay, so as I said, in our last lesson, we looked at carbohydrates. We looked at the majority of the spec, but we didn't have time for this final part of the spec, which was biochemical tests using Benedict's solution for reducing sugars and non-reducing sugars, and iodine potassium iodide for starch. Okay, that's what we are going to be looking at in today's lesson. So we're going to start off by looking at the test for starch. Now, this should be a nice, easy recap from GCSE. Okay. So test for starch. These are the two main points that you need to be familiar with. You have to add iodine into a food sample solution, and then you would get a positive result, a color change from orange to blue-black. Okay, it's the same as GCSE. Okay, and I've highlighted the key points there. You have to add the iodine, and then you have to observe the color change from orange to blue black. Okay, normally this is done by getting the food sample. You will normally have a test tube such as this. You would dissolve your food sample, and then you would add in your iodine into this food sample, and then you would observe the color change. Okay, that's what you would not substance into each tile, and then you add different concentrations of iodine to observe the color change once again. The key part over here that you need to understand is the process of doing it. You have to add iodine to a food sample, and for a positive result, you would observe a color change from orange to blue-black. Okay. Now, that's the test for starch. Moving on to the test for proteins. Now, this is again nothing new. This is still GCSE. We've not moved into A level yet. You have to add burette solution, and a positive result will change the solution from blue to purple. Okay. So again, very similar process. You would normally have a test tube with your sample. You would add in your burette solution. To the test tube, and then what you realize is that this will change color for a positive result. Okay, just be familiar with the colors. Starch is orange to blue black, and proteins is blue to purple. That's the main part that students get confused with. So just make sure that you've learned the color changes, because that was probably the hardest part of GCSE, and believe it or not, that's the hardest part of the A level for this part of the spec. Okay, so we've done starch and we've done protein. Starch, iodine, orange to blue black. Proteins is burette, blue to purple. Okay, now for lipids, it's the same as GCSE. However, the mark schemes for this question have a specific order in which they want you to answer the question in. Okay, and I've highlighted the points over here. First of all, you need to dissolve the sample in ethanol. Okay, this is the first part. And then you add distilled water. Now, the reason why I've highlighted both of these steps is because normally people get confused in the order. You have to have this order, or simply the test won't work. Okay, if you had a test tube like this, and then you added distilled water first into the test tube, and then you added ethanol, the test won't work. You have to add in the ethanol, then you add in the distilled water, and then the test will work if a lipid is present, obviously. Okay, so just remember the order. You need to add in ethanol, then water. Think of it like this. Whenever it comes to remembering the order of something, I look at the first letter. E comes before W, so we're lucky there. Dissolve sample in ethanol first, then water, because E comes first in the alphabet and water comes after. And then again, you will always have a mark for the positive result, and the positive result is a white emulsion. Now, this is your safest sort of way of presenting the positive result. People say cloudy, people say precipitate, but emulsion will be present on mark scheme. So just say emulsion. Don't say cloudy, um, precipitate, because mock schemes will reject that word. It wasn't much focused on at GCSE, but at A level it is focused on. So make sure you say emulsion and not precipitate, or milky or cloudy, or anything like that. Just say white emulsion. It shows the examiner that you know what the positive result is, and you know exactly what the correct answer is. Okay. So I think out of those that we've looked at, lipids is probably the one that students get wrong most. So make sure you learn this process. Obviously, the other two, starch and proteins, are basically the same, exact same as GCSE, and you wouldn't have had that many misconceptions with them all the way through to A level. Iodine, blue black, burette, purple. However, lipids, there's a process. Ethanol, water, white emulsion for a positive result. Just remember the process. You need to add in ethanol first, then you add in water, and for a positive result, you will get a white emulsion. Make a flashcard of this. This is a model exam answer. Okay. Now, after lipids, there's one more, which is reducing sugars. And in terms of reducing sugars, this is actually, we've looked at GCSE as well. The test for sugars, we've looked at the generalized test for sugars. Um, and reducing sugars is the generalized test for sugars. Okay, so the process is still the same, but there's one new part from A level, and that's the examples of reducing sugars that we have. So a reducing sugar, you add in Benedict's reagent and you heat. Now this part people forget. Normally this question is worth 3 marks. So if you go back, this question, lipids, is normally worth 3 marks. And this question is also worth 3 marks. And the reason why is because it's got three key marking points. Um, Benedict's reagent, heat, and brick red precipitate. You will always have 1 mark for the result. You will have 1 mark for the reagent. However, in lipids, you have 2 stages where you have to add in ethanol, then water, and it will always say needs to have correct order, or you will get it all wrong. Okay, so there are your 3 marks for lipids, and here are your 3 marks for reducing sugars. You have to add in Benedict's reagent, and you have to heat the solution, and only after you've heated it, is when you'll receive the brick red precipitate. Okay, so those are your key points. Now, last lesson, we looked at carbohydrates and we looked at monosaccharides and we looked at disaccharides. Okay, now what you would have realized that is that this topic also has a relationship between the monosaccharides and the disaccharides. Remember in our first lesson, we looked at monomers and polymers. Okay, and here I'm just doing a quick recap of everything we've looked at. This is a polymer. Each circle is representing a monomer. The whole chain over here is known as a polymer. And we looked at an example for carbohydrates, which you are expected to know. Carbohydrate is an example of a polymer, and it's known as a polysaccharide. Okay, now because saccharide has poly in front of it, that must mean each individual monomer is a saccharide. Each individual monomer is a saccharide. And we looked at carbohydrates, we looked at different examples. We looked at starch, cellulose, and glycogen. So e.g., we looked at starch, we looked at cellulose, and we looked at glycogen. And we also looked at some examples of monomers, the monosaccharides. So we had monosaccharides, we had glucose, we had fructose, we had galactose, and etc. These were some examples of monosaccharides. And when these two monosaccharides come together, for example, two glucose molecules come together, we saw that a condensation reaction happens within these two monosaccharides to form maltose and water. Okay, now a condensation reaction means the formation of a glycosidic bond, a chemical bond. In terms of carbohydrates, you've got a glycosidic bond being formed, and then a molecule of water is being released in the process, and you're forming a disaccharide, which is maltose. You've combined these two glucose units to form a structure which has two glucose units. That's why it's called a disaccharide. Di means two. And then in doing so, you formed a glycosidic bond, releasing a molecule of water. So that's what we looked at in our last lesson. Just a bit of a recap there. When you're testing for reducing sugars, you're also required to know some of the examples of reducing sugars. And here are some examples of reducing sugars. I'm just going to get rid of some of my writing. Okay. So examples of reducing sugars include just a minute. Okay. Examples of reducing sugars include glucose, fructose, galactose, lactose, and maltose. Okay, so those are just some examples of reducing sugars. Well, if I show you the example of a non-reducing sugar, then you will realize that there's only one example of a non-reducing sugar, the rest are reducing sugars. Okay, so remember when we looked at chemical reactions, just a minute. Okay. When we looked at examples of reducing sugars, we looked at some chemical reactions, and that was glucose plus glucose produces maltose plus water, and then we had glucose plus fructose produces sucrose plus water, and then we add glucose. That's really bad writing, sorry. Okay. Okay, and then we add glucose plus galactose produces lactose plus water. Okay, those were some of the examples of reactions that we looked at. And as you've realized, we had basically all our salt reactions. You have glucose, glucose, we have maltose, glucose, and maltose here. We have fructose here. We have sucrose is something we don't have. Um, and then we had galactose and lactose. Okay, so as you can see from this list, there's only one thing that we don't have. We don't have sucrose. And that's because sucrose is not a reducing sugar. Sucrose is a non-reducing sugar. Okay, so whenever you're asked about sucrose in an exam, you need to know that sucrose is not a reducing sugar. Okay, just bear that in mind. Okay, so before we move on to the new A-level content, which is just another process that you need to be familiar with, what you need to know is what we've all looked at and be confident with that. First of all, test for starch, iodine, orange to blue, black. Proteins, add burette, blue to purple for a positive result. Lipids, add ethanol, then water, and you will receive a white emulsion for a positive result. And then reducing sugars, add Benedict's and heat. A brick red precipitate will form for a positive result. And examples of reducing sugars include glucose, fructose, galactose, lactose, and maltose. Okay, and I'm just going to pause there for you guys to make any notes of that slide, and then I'm going to move on in 30 seconds. Okay, so hopefully you guys have had enough time to make any notes from that slide. Moving on now. Okay, this is the new part of the content, and there's only one slide. There's not that much new. It's just a process that you need to be familiar with. And then we're going to move on to some exam questions to finish off for the session. Okay, test for non-reducing sugars, and this is the new A-level content. And the example that we are going to be using is sucrose because that's the one you need to know for the spec. So first of all, you have to obtain a negative Benedict's test where the reagent remains blue. Okay, so for example, we're going to have our test tube. We're going to have our sample in the test tube dissolved. Okay, we're going to carry out a normal Benedict's test. Okay, and how we're going to carry it out, we're going to add in Benedict's reagent. So we've got Benedict's reagent, which we're going to add, and then we're going to heat. After we've heated, if we've not seen a brick red precipitate occur and the solution has remained blue, then we're going to carry out the following process. So we've done a Benedict's test and we've not had a positive result. Okay, what next? Well, next you have to add in acid and boil. And the reason for this is for the acid, for hydrolysis to occur. Now, remember what hydrolysis is? Hydrolysis is basically using water um, and splitting the molecule up. Okay, so you're going to split the molecule split the molecule, which in this case is the disaccharide, which is sucrose. And in doing so, you're breaking the glycosidic bond. And you're releasing the monomers, which is ideally your glucose and fructose, if we're talking in terms of sucrose. Okay, so we've done a negative Benedict's test. The reagent hasn't changed color. Now we have to add in acid and we have to boil. And the reason why we are doing this is basically to make sure that the sucrose, which is a non-reducing sugar, the glycosidic bond between sucrose is broken. And if the bond is broken, what's going to happen is that the individual monomers, they're going to spread out. Okay, they're going to come back into the solution, glucose and fructose. And what you would realize is that both glucose and fructose are examples of reducing sugars. Both are example of reducing sugars. And if they are examples of reducing sugars, that must mean that they should now show a positive result for the Benedict's test. Okay, so we've added acid and we've boiled. Okay, we've split the sucrose into its individual monomers. Now what we're going to do is we're going to cool the solution and then add in alkali to neutralize it. By the way, if you want an example of an acid, we normally use HCl. And in terms of alkali, we're going to use sodium hydroxide. Okay, we're going to cool the solution and then we're going to add in alkali so we can neutralize it. And the reason why we are doing this is because Benedict's doesn't work under acidic condition. Okay, so the reason why we are doing this is basically because Benedict's doesn't work under acidic condition. Doesn't work under acidic conditions. Okay, it needs to be neutral for it to work. That's why, because we've added in acid over here, we're going to add in alkali over here so we can neutralize the solution. So it's not either acidic or alkaline. Okay, and because we've boiled it over here, we're cooling it over here. We're literally bringing it back to its normal conditions. So you've added your acid and you've boiled it, you've broken the bond and you've released the individual monomers, which will show a positive Benedict's test. Now you've cooled the solution to add in alkali to it so you can neutralize the solution and bring it back to its normal pH. Now this normal solution, this solution that we have, no longer has sucrose, it has the individual monomers of sucrose. So now it should show a positive result when we add in Benedict's reagent because it has glucose and fructose present in solution. So now what we're going to do is we're going to follow the normal Benedict's test. We're going to add in Benedict's reagent and we're going to heat. Okay, we're going to add in Benedict's reagent and we're going to heat. And this is the same exact process that we looked at on the previous slide. Okay, so you add Benedict's reagent, heat, and then you would see a brick red precipitate for a positive result. Okay, so this is the same as tests for reducing sugars. Okay, So that's basically the test for non-reducing sugars. First, you need to make sure that you don't have a reducing sugar present. So you obtain a negative Benedict's test result. And then you have to focus on breaking the glycosidic bond. So you have to add in acid and then you boil. Okay, so the hydrolysis can actually occur. And if we're talking in terms of sucrose, the sucrose can be split into its specific monomers, monosaccharides, glucose, and fructose. Now, because we've added in acid and we've boiled, we need to bring the solution back to its normal conditions. So we're going to cool the solution, and because we had added acid, we're now going to add in alkali. And the purpose of the alkali is to neutralize it because Benedict's test will not work under acidic conditions. And now you brought the solution back to its normal, you can then add in Benedict's reagent and then you can heat the solution once again. And then if you see a positive result, which turns brick red, which forms a brick red precipitate in the solution, then you know that originally you had a negative result. Now you have a positive result after adding the acid in. So you must have a non-reducing sugar. Okay, and that's basically the A level part of biochemical tests. Again, I'm going to pause there for a minute or so to answer any questions. I can see some coming in the chat. I'm just going to take a moment to answer them while you guys make any notes of the slides or ask any questions you have in mind in the Q&A. Okay, guys, I think I've answered all the questions which have come through. And now basically that's the end of the content that you need to be familiar with on biochemical tests. And now we can move on to answering some exam questions. Okay, if anyone has any more questions, please just keep letting me know in the Q&A and I'll keep answering them as soon as I can. But we're going to move on now to looking at some exam questions.